Thank you, thank you. TED Talks are supposed to be about having one big idea, and because we have a short time to speak. And believe it or not, the big idea that I'm going to tell you about starts with corn, and it starts with blue corn, Indian corn. And it's sort of appropriate this time of year. If you go into the produce section of supermarkets, you'll see lots of bunches of ears that look very much like this. So in fact, what I'm going to tell you about is about a person who is really my hero in science, a person that, whose footsteps I'm following in. And so I'm going to sort of tell you the background to what we actually do in our lab at Riverside. And as I said, it starts with corn. No. All right. So the question really is, the question I'm asking here is why is corn blue? And I've sort of stripped it down to the basics. And that is that there are genes. We all have genes. All organisms have genes. Genes make protein. Proteins make us, basically, and make all the organisms, all the plants and animals. So in this case, we're going to focus down to one gene. And that's a gene that makes corn blue, makes the kernels of the corn blue. So we have this gene. When it's working, we have blue corn. Okay? Simple. We eat this kind of corn. We eat yellow corn. All right? So it's different. Okay, so how is it different? It's different because that gene's not working. So in fact, we eat mutant corn. And I haven't really figured out why the breeders bred it, the color out. But you might imagine that with the color in, you know, you get corn stuck between your teeth and it wouldn't be colorless, it would be blue. I don't know, but most of the corn we eat is colorless. Although I guess in organic stores there is blue corn, and it's not any better for you. The blue is not good for your health necessarily. It's not bad for you, but it's not particularly good for you. So I've told you already about the colorless corn, yellow corn, and the colored corn. Now here we see something very different, spotted corn. And that's really where this big idea comes from, or where it starts. And that is, so I've told you that when the gene is working, the color gene, we have colored kernels. When the gene's not working, when it's mutant, when the gene is mutant, has a mutation in it, we have colorless kernels. So the question is, how do we have spotted kernels? Okay? The person who solved that problem is this woman, Barbara McClintock. And she really is the person that is my inspiration in science. And I'm going to tell you a little bit about her. You see her here, picture with her holding Indian corn. So here's another picture of Barbara McClintock. This is from 1929, and it was at Cornell University. And she was part of a group of corn geneticists, people that worked with corn, much like people work with fruit flies or, I don't know, E. coli now. She worked with corn, and her professor was here, R.A. Emerson. So that's Barbara. You could see also she's wearing knickers, all right? Women didn't wear pants in those days. They wore skirts. But she grew up, she was a tomboy. She liked to climb, she did a lot of things that typical girls and women of that time didn't do. And her mother actually made her clothes, so she was able to wear pants like here. I'll show you in a bit the only time I've ever seen her not wearing pants, but that was for a very special occasion. So anyway, what is corn genetics? And this is really simple also. You see that the people over here have strange things on them. Those are actually paper bags. And what you do, genetics is about crossing things. It's mating. It could be animals. It could be when you mate plants, you cross them. We say we cross them. So it's basically taking the pollen from one plant and putting it onto the ear of another plant. So you're controlling the cross, and corn is really nice for this. And that's what these bags are for. These bags are so they can put it on the top of the corn plant and collect pollen. So that's corn genetics. That's the technique that Barbara McClintock used to solve this spotted corn problem. So again, here she is, younger than in the previous picture. Here's in her cornfield holding the bags. And the question she asked was, why are these kernels spotted? And here's a page from her notebook. This is when this was actually typed out in the old days, and you could see that she looked at these kernels, she came to, she asked questions, she asked why is it this way, she did crosses, and so finally, after many years, she came to a conclusion, and that's shown in this slide. So essentially, with this seemingly trivial thing that she's dealing with, why are kernels spotted, she actually discovered a unique component of chromosomes of the genome. So the question, she discovered a new type of mutation. So that mutation is, I'll show you this color gene again, and that mutation is caused by another piece of DNA called a transposable element, and it's a piece of DNA that's in this case, the corn chromosomes, and it can move around. And so in this case, it jumps into the color gene, but just as it jumped in, it could jump out, it could come out. So unlike the other mutations, so you might have a mutation that's a piece of DNA is torn out, it's deleted, it's permanent. This is not a permanent change, it's a temporary change. And so that's what we're seeing here. And that still probably doesn't sound like too important, but we'll get to that. So to explain the spotted kernels, what McClintock said is that when you see the kernel and you have this yellow area here, that in fact is a cell, many cells, that have in them, in their chromosomes, a gene that has a transposable element stuck in the color gene so that there is no color. However, in other cells in the kernel, that transposable element can jump out and restore the expression of this gene so that you can get colored sectors. Okay, so that's as hard as it gets. But it still doesn't sound very important, right? So, but what she in fact discovered was a new component of chromosomes, of our genome, of all genomes. So here's a view of the chromosome before McClintock. And these boxes would be genes. So it would be gene, 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 gene. Just the whole thing would be genes. What she discovered was, in fact, there's a new resident, there's a new component, a different component in the genome. And that I've drawn as these little ellipses, and those are the transposable elements. So she discovered a component in this case, the corn genome, okay? But the significance is that transposable elements are not just in corn, okay? And they are, so after 40 years, she discovered this in the 1950s, a long time ago, okay, when I was born. So she discovered, over the years, it was found that these are in virtually all organisms on this planet. In the 1950s, transposable elements were found in the fruit fly and Drosophila. In the 1960s, they were found in bacteria, in E. coli. And finally, in the 1970s, they were discovered, and TEs is a shortened term for transposable elements. In the 1970s, they were discovered in humans, okay? So this is a picture of a rose from Sonoma in the wine country. I was sitting on a bench, and I looked behind me, and there was this beautiful rose. And this is whoops. And this is a rose that where there's also a gene that has a transposable element in the pigment gene. Here's a petunia flower with also the pattern characteristic of a transposable element moving around. So the transposable elements have other names that you may or may not be more familiar with. They're called mobile DNA. They're called jumping genes. And finally, in a derogatory sense, they're called junk DNA. All right? So this is a picture of Barbara McClintock, again, wearing, in this case, a gown. It was a very special occasion. She's receiving the Nobel Prize in 1983 in medicine or physiology for her discovery of transposable elements. Okay? So as I said, this is probably one of the few times that she wasn't wearing pants, because it is a pretty special occasion when you get to meet the King of Sweden. So the question that really, so this still isn't really a revolution. What's so revolutionary about that? And that's the part I'm going to tell you in just maybe 3 or 4 slides. So let's ask how much of our chromosomes are due to transposable elements. How much transposable elements are in us? And the example I give, so our chromosomes have, our DNA, our chromosomes, are made up of building blocks. Those building blocks are A, G, C, and T, the bases. I'm sure you all are familiar with that. That's all even taught in high school biology. So there are 2.5 billion letters in our genome, in our chromosomes, okay? That's equivalent, and these are textbooks here, some of them, well, you're familiar with textbooks certainly. This is equivalent to 1,000 textbooks of 1,000 pages long. No pictures, no anything, just letters, okay? The genes that make up us, that make up take up only 20 to 40 of those textbooks. That's it. All of the information that encodes all of the proteins and everything else that makes up us fit into 20 to 40 textbooks. 500 of the textbooks come from transposable elements, okay? Half of our genome, half of our chromosomes is this stuff that was discovered by Barbara McClintock. So what I want to say is that transposable elements are really the largest component of the chromosomes of all of the higher life on this planet, all plants and animals. To give you an example, I already said that about 50% of the chromosomes of humans, mammals, humans, apes, and mouse are from transposable elements. 75% of the corn chromosomes are from transposable elements, derived from transposable elements. And amazingly, irises, beautiful flowers, 98% of their chromosomes are due to transposable elements. And there's iris. I mean, it's a beautiful plant, beautiful flowers. You know, who'd have thunk, right? So the questions, I mean, there's lots of questions that come, and that's why there's a wonderful area to be researching in. Because you look at this, and it's sort of the other end of the universe that you heard about before, but it's on a small scale. So it's first of all, how do we in all life live with all this stuff that's far more predominant than our genes? And one reason is that most of it is dead. Most of it is accumulated, all sorts of mutations, so the DNA is there, but it can't move around. It's just disappearing, really, over millions, hundreds of thousands, millions of years. So most of it is dead. The ones that are alive, that can still move around, move into places that it won't harm the organism, and I'll show you an example of that in the next to last slide. So they go into what we call safe havens. So these little pieces of DNA, their sole goal in life is to make more copies of them and just be carried along with all the genetic information. And they're doing a very good job of it. But they can't kill the organism, because if they do, they're gone too, because they're us. So the other thing is organisms have what could be referred to as chromosome immune systems. Organisms can identify these transposable elements in their chromosomes, in their genes, and shut them down, okay? So this is just an example, one example of many. This is a region of the corn chromosome, of one corn chromosome. These are the genes. These are the transposable elements. And this is typical. So I said for something to be 50 to 80 to 90% of the genome, like this is sort of one way you could, not one way it might look, this is one part of a chromosome in corn. So the question that I'll sort of leave you with is, are transposable elements useful? That's a question that we're asking in my lab and in many labs around the country. So it's probable that most of it is junk, okay, which is pretty amazing. When I learned about biology when I was in college a very long time ago, we sort of learned that living things were almost perfect. They were, you know, these really well-oiled machines, and everything worked perfectly. No, obviously everything didn't work perfectly, but everything worked well, and there was little waste or whatever. It turns out that most of our genome, when we look closely under the hood, it's not like that, okay? So most of our genome is probably junk, but some of it generates genetic diversity, okay? And this genetic diversity helps species to adapt to a changing environment. And so that, as I say, is for another TED Talk. That's another big idea, so I thought I'd leave you with that. Since it's the last talk, I didn't want to tax all of our brains. And really, so when you're out there in the produce section, looking at the corn, you may look closely. Some of it may be striped, some of it may be spotted, and give a toast to Barbara McClintock. She was an absolutely remarkable woman. Like I said, I had the good fortune of knowing her for the last 10 years of her life. She died in 1992, and she really is a giant in genetics. And there's books about her, and I would encourage you to look online. To me, it was the most wonderful thing in my life, scientifically, to know somebody like that. Because in biology, she's sort of in many ways equivalent to an Einstein. So I'll stop there, and I'll be happy to take questions.
Okay, we've got some time for some questions. If anybody has a question for Dr. Wessler, I do have a mic here.
Yes. One over there.
The transposable elements in humans, are those the freckles?
I missed the end.
The humans that have transposable elements, are those the freckles?
Oh, all humans have transposable elements, whether you have freckles or not. And freckles aren't due to transposable elements, right? But it's a good question. They're not, yes. They're actually, you know, if you go in the sun, freckles will come out. So a lot of it is environmental, it's not your genes, right? Other questions? Yes. She's asking, what are some of the effects of transposable elements in humans? Transposable elements have been documented to cause some genetic diseases, one of them being hemophilia. So hemophilia is caused by mutations that are in genes that are responsible for, what is it, hemoglobin? See, I'm a plant biologist, so I don't know all this. But anyway, so there are examples of transposable elements causing human genetic diseases. But for the most part, our elements don't move around. So one element may move for every 200 human births. So you don't, even though our genome is 50% transposable elements, most of it isn't going anywhere, okay? Virtually all of it isn't going anywhere in all of our lifetimes. Like I said, once in every 200 births, one of these elements moves someplace else. And now that they're sequencing human genomes, hundreds of them, we can actually see differences due to movement of transposable elements.